Hello everyone. In our previous lecture, we have discussed about the quantum of data that needs to be generated for an RNA-seq or a DNA-seq experiment. Now, we'll see how we can counter-check whether we have generated the right quantum of data or not. Per se, the vendor is asked to generate 80 million reads, like what you have calculated for your whole transcriptome experiment, 80 million reads. And you need to counter-check whether your vendor has generated the right quantum of data or not. So the first thing that you'll have to understand is the format in which the data is supplied. The format in which the data is supplied is FASTQ format. Now, for us to understand what FASTQ format is, we'll differentiate FASTQ format from the FASTA format. What is FASTA format? You have a greater than sign followed by a FASTA command line. And then you have your sequence in the next line, whatever is the sequence. Now, when I write this specifically, this means that the sequence has to be entered in the next line. And this FASTA command line includes all the metadata of that particular sequence. All the metadata of that particular sequence. Now, it means that if you have a sequence like this, let us say, after the FASTA command line, if you are starting your sequence, this particular sequence is not recognized to be your FASTA sequence. Right? Now, we'll see how this is different from the FASTQ format which you are going to actually get from the vendor. The FASTQ format, the sequence would start, instead of greater than sign, it would start with @ sign, followed by, you'll be having a line here, and this is the FASTQ command line, which contains the machine ID, and then followed by the X and Y coordinates, followed by 1 or 2. I'll explain everything. And the next line would be your read that is generated. For example, here 80 million reads we have studied, we have generated for 2 into 150. So this 150, this particular read length should run to 150 bases, And the next one will be a plus sign and followed by the last line, which contains the quality. And then starts the next read again, followed by @ sign, again the same machine ID. And now let us say, to differentiate this read from the next read, what we do is, let us make it as X1, Y1, and this will be X2, Y2. And this will be either 1 or 2, followed by some sequence CTAGC running to a length of around 150 bases again, followed by a plus sign and followed by the quality of each and every base that is being read in this last line. So what does this mean? Means each read that is generated will be covering 4 lines. Each read that is generated covers around 4 lines. It means that if you have generated 80 million reads, how many lines will the file contain now? The file will contain 80 into 4. That makes it a total of 320 million lines. So the moment the vendor supplies the data, the first thing we do is we immediately cat the file in Linux or Mac. Means we cat stands for opening the file, and then the file name, then pipe it along with WC minus L. Means word count in terms of lines. This will immediately throw a 320 million here. on the screen, then it means immediately for this file name that you have given here, you have 320 million lines. It means that there are around 80 million reads. So that's how you initially counter check your data for the number of reads that are generated in a file. Now, the next thing that we'll have to understand is each and every line of the FASTQ format needs to be understood. We'll have a look at each and every line and then understand what it means. So we'll go back to this here. Now, what did I mention here? The line will give a machine ID. For example, if you have generated your data by using Illumina Hi-C, right? Or Illumina Nova-C, sorry. MyC or Nova-C, then that specific machine ID will be given here. And this will be common for all the reads. This will be common for all the reads. That's what we need to understand. Okay. Now, the next thing. What are these X and Y? You should understand what are these X and Y and what is this 1 and 2. Now, let us see. What did I tell you initially? When you have a fragment, that's what we explained. When you have a fragment, right? You have an adapter on one side, you have an adapter on one side, and you have an adapter on the other side. And you have a primer targeting the adapter from the forward direction and also primer targeting from the reverse direction. Now what happens here is, as your sequencing is going on in the process of synthesis, this is also called as sequencing by synthesis. So what is happening? If the template has got a G, you will be having a C added. And if it has got T here, you'll be having a A added in the process of synthesis of this particular read. And this read here runs to a length of around 150 base pairs, for example, right? Now, what happens? This C that is being read here is basically attached to a fluoropore. Now, this fluoropore is read by the CCD camera, which is there. And depending on the color, it will recognize and it will see whether C is added or a is added, right? Now what happens? If you have a single fragment, and one C is added, and the quantum of light that is emitted from this fluorophore from a single C added will not be sufficient to be recognized by the CCD camera and then finally read as a C, right? Once again, I'll repeat. If you have a C added, and if you have a single C that is added, and this single C, the quantum of fluorescence that is emitted by a single C will not be enough to be read by a CCD camera and to be recognized as a C that is there in the process of sequencing. This will not be recognized if it is a single C. So what do we do normally? There is now this particular fragment that you have this fragment, right? To this fragment is now amplified in all the platforms, right, that you have, that will be Illumina or the solid platform, you have an amplification step involved. So what do? This particular fragment now with the adapters here is now amplified, And when this is amplified, what happens? This particular fragment having G here will be there in many fragments now. So we'll draw this fragment after amplification as a single line. So you'll have so many fragments. Each fragment, though I'm drawing a single line, is a double-stranded molecule. Now, what do you have here? And each one of them now has got an adapter, which I'm drawing with a red line here, right? This is a red line on the adapter on either sides. Now, when sequencing is taking place, now let us say, once the same fragment is amplified several times, and since you have got a G everywhere here at this particular point, and now when the sequencing takes place, C is added here, and when there is G here, C is again added here, C is again added here, C is again added here, and C is again added here. Earlier, you have a single fragment and a single C added. Now what happened, since you have amplified this fragment and you have several fragments of the same type, and when this synthesis takes place and C is added, there are several C that are added. And now this fluorescence would be enough for us to be recognized by the CCD camera, and it will be read by the camera as C. Now, what does this mean? Means all the fragments that you have fragmented initially will undergo amplification on the platforms, especially Illumina and Solid, not in PacBio and Nanopore, right? Just remember, only in Illumina and Solid. Now, you know that there is amplification that is taking place. Now we'll have a look at how the amplification takes place in Illumina. In Illumina, you basically have a flow cell. And on the flow cell, you'll be having lanes. I would request you to go to YouTube and watch the video on bridge amplification. Right. If you watch this video, this will be much more clear. Now you'll be having lanes here. This is one lane. I'm drawing only a flow cell with 2 lanes. You can have a flow cell with 4 lanes as well. And you'll be having lanes here. And on these lanes, now I'll magnify these lanes. Let us say part of this lane will be magnified. This particular part I'm taking, and I'm magnifying here, right? So this particular part I'm taking and I'm magnifying it. Now, look at this. This looks like a square that I've taken from there. Now on this, there'll be several probes that can bind to your adapters, which I was talking about, right? So there is a region here, let us say, where you have your fragments amplified. Means what happens here, we'll further re-magnify this. Means if you take a spot here, spot will contain a probe, right, which will be complementary to your adapter that you have attached to your fragment. This will be complementary to your adapter that you have attached to your fragment. So now your fragment is here, right? And it contains adapters, and this will be complementary, and it will bind to this particular spot. So there is a mechanism wherein at this particular region, this limited circle which I'm showing you, once you are actually attaching your adapters here, there will be probes that will be complementary to your adapters and will bind to your adapters here. And then we'll bring about an amplification step, and that is called as bridge amplification. So what do I mean here? Means if you pick up a spot here, you will be having all the fragments that are there here of similar kind. So what does this mean? All the fragments that you have in the near vicinity here will be of similar kind, and they'll be around, let us say, a minimum of around 10,000 fragments. Means you have taken your original fragment and you have allowed your original fragment to go and bind to a sequence that is already there on your lane, standing upright. Means you have a lane like this, and you already have a adapter sequence that is complementary to your adapter. You have a probe sequence that is complementary to your adapter, and this probe sequence will bind to your adapter. and will undergo bridge amplification. And once it undergoes bridge amplification, what happens? In the near vicinity, you will be having all the fragments that are of similar kind. So in the process of amplification, like what we had shown earlier, when a C is added to all these fragments, this C now will be recognized by your CCD camera, just because there will be several C that will be enough to generate the quantum of fluorescence that can be recognized by the CCD camera. Once again, you have a flow cell. On the flow cell, you'll be having probes against your adapters, and these probes will bind to your adapters, and an amplification step takes place. And that amplification is called as bridge amplification in the case of your Illumina. And you have a spot here which contains all the fragments of similar kind. And when amplification takes place at this spot, then C is added and this C is recognized by your CCD camera, right? Now, we'll go back and see how the data looks like for this particular fragment. Now you have another spot here. There'll be a second kind of fragment that is generated here, right? There'll be a second kind of fragment that is generated here, because there'll be some other fragment that is attached to your adapters, and there is bridge amplification. There's a different kind of fragment here. And similarly, there'll be another spot here. See, though I'm drawing bigger circles, they're quite close to one another, and you'll have all these bridge amplification within that closed region for that particular fragment. And the amplification is simultaneously taking place for all these regions. And similarly, you have a region here, and again, you'll have fragments of similar kind. You'll have a region here, you'll have fragments of similar kind. Now, let us say that now the data is generated. Now the data is generated from this one, and you have allowed your forward primer to bind. You have allowed your forward primer to bind, and the amplification has started to take place. So we'll see how the read is generated from this particular spot. The read will be, you'll have @ sign followed by the machine ID. Now what happens? This particular read, this is the X coordinate and this is the Y coordinate. Means if you say that this is X1, and if you say this is Y1, right? Means what is this? The coordinates, if you see X and Y, the spot is here. So it means it says the machine reads this particular C that is being added here in this line, right? It has got an X1 and a Y1 coordinate, X1 and a Y1 coordinate, right? And then if there is a, let us say for a second read of this particular, let us say we'll take a second read from here, and you have @ sign again here, the machine ID will remain the same. Now have a look at this. For this, the Y1 will remain the same. For this, the Y1 will remain the same. But what's happening here? The X1 will now become an X2, because this is an X2. The place where the coordinates for this will be an X2. So this read will be an X2 and a Y1. So now let us say from this, you get A that is being read. And similarly, if you are looking at this particular spot, we'll take a spot from here, and this is the @ sign. And now you have a machine ID here, followed by, now what do you have? You have some Y2 here, and this is X1 here. Now you have a X1, Y2 here. And the read, let us say that you're reading here, you're getting a G from this spot. All this is taking place in one single go, right? And that's why this is a high throughput data generation. And once you have all this in one single go, there is every possibility that there's a lot of background signal. And that is the reason why you need to have quality attached for each of this that is being read. And that's why in the fourth line, you have the quality attached for each one of this that is being read, right? No. So now what do you understand? We have understood that after bridge amplification, a particular spot will be having fragments of similar kind, and all these fragments would generate a signal that is enough for us to be recognized by the CCD camera. And the different spots will be having different X and Y coordinates. Now, after C, let us say there is after C here, you have a, let us say, on the original fragment here that you have a here. Now what is added here? It's T added. Now it is T added in the amplicon. Now when T is added in the amplicon, right, the T comes from here, right? So whether the T is coming from this or from some other place, let us say here as well, there is a T coming next, right? The T coming from this spot will be added to this X and Y coordinates. And the T coming from this spot will be added to this X and Y coordinates. So the X and Y coordinates will actually determine how your read entirely is being read from a particular address. So what is that address? The address is nothing but your X and Y coordinates, right? If you have understood X and Y, fantastic. We'll next go to the next one. What we'll do is, we'll see this. Since you have understood X and Y, the next thing that we need to understand is, I told you something about 1 or 2, right? Now, from a particular spot, you can get both your forward and reverse, paid and reads, right? You can get a forward and reverse, paid and reads. So in a file, let us say, if this is the forward read, that is designated as 1. Means if you have a 1 here, it means definitely having got all the same coordinates, right? You will definitely have a mate of this in the file present. Now, what do I mean by that? What do I mean by that? We'll go back and have a look at what I meant. Means, now let us see. Now, for example, like you say that you have got a forward read for this, and the forward read is designated by 1. And now reverse read for the same, fragment, for the same fragment, I mean, right? Reverse read for the same fragment will be having the same machine ID and will also be having the same X and Y coordinates because it's generated from the same lot. And now, how will the reverse read be designated as 2? 2 means, once again, you have a fragment, right? This fragment has got an adapter A1. This has got an adapter A1 here and an adapter A2 here. And this is the forward direction and this is the reverse direction. The forward direction read is given here, whereas the reverse direction read is given here. Everything else is same, but you'll be having a 1 and 2. So what have you looked into here? You have got a read that is generated in the forward direction from the same fragment and a read that is generated in the reverse direction from the same fragment. Now, this is how you make out in your file by looking at the 1s and 2s having got all the other coordinates similar, right? Whether this particular read that is there in the file has got its mate or the paired and read that is there for this particular forward read. And this is what we understand by the FASTQ file that we look at. Number of reverse and the forward reads by the number of 1s and 2s for the corresponding reads that are generated. So in your experiment, if you have generated 80 million reads, you expect that there'll be around 320 million lines, the first thing. And then by means of these 1s and 2s, you should be having 40 million forward, means once, and 40 million reverse, that is twos. Thank you.